Hello and welcome to today's ClinData Insight webinar, Let's Make Oncology Studies Easy for Programmers and Statisticians. This webinar is hosted by ClinData Insight. My name is Mylyn Palieri and I will be your moderator for today. Our presenter is Kevin Lee. ClinData Insight is a biometrics consulting firm based in the San Francisco Bay Area. We specialize in biostatistics, statistical programming, CDISC implementation, big data for life science, and talent solutions. Today, our topic is Let's Make Oncology Studies Easy for Programmers and Statisticians. Kevin Lee is the Director of Data Science at ClinData Insight. Kevin has successfully led many oncology studies as a project lead programmer, statistician. Kevin is known as an oncology expert in the industry, helping and teaching many programmers and statisticians on oncology studies, response criteria, CDISC implementation, data collection and analysis, and FDA submission. Kevin has presented more than 50 presentations, including more than 10 oncology-specific presentations. Kevin's favorite topics include oncology, standards, and Philly sports teams. Before we begin, first I would like to go over a few general housekeeping announcements. So from now until the end of the webinar, all attendees will be placed on mute so that we can remain on track. Unfortunately, we are unable to control the ding, as you can probably hear when people are joining us, but we'll try to keep on moving on. If you have a question at any time, please submit them via the question box located at the top left-hand corner of your webinar screen, or you can even include your questions down at the bottom in the chat box at the lower left-hand corner of your screen, and I'll try to keep an eye on that box as well. Questions will be answered at the end of the presentation in the order received. And in addition, we will be recording this webinar and later have it available on our website. And if you're familiar with our YouTube channel, you can find it there as well. We estimate the webinar will end in about 45 minutes from now, but if there are a lot of questions, we can stay a little longer to get those questions answered. But if we do run out of time at the end of the hour, to be mindful of everyone's schedule, any remaining questions will be answered offline by Kevin, and the answers will be placed on our website along with the slides. With that, I would like to welcome Kevin and hand over the controls. Kevin?
Thanks, Martin. Can you hear me?
Yes, I can.
Great. Thank you. First of all, thank you all for joining our today's webinar, and good morning to those in West Coast, and good afternoon to those in East Coast. Thank you all for joining our webinar today on Let's Make Oncology Study Easy for SAS programmers and biostatisticians. Our agenda, we're going to start with oncology introductions, and then we're going to introduce oncology subtypes such as solid tumors, lymphoma, and leukemia. And we're going to also talk about their response criteria guideline and also oncology-specific data collection such as CDISC data standards, and also data analysis such as overall survival and progression-free survival. And then we're going to conclude our webinar with the final thought and Q&A. So let's talk about oncologies or cancers. The word cancer is related to the Greek word crab because its finger-like projections were very similar to the shape of the crab. In year 2010, economic cost of disease is worldwide was estimated about $1.2 trillion. And 1 in 8 deaths in the world right now is due to cancer. WHO predicts that new cancer cases will increase from 14 million in year 2012 to 22 million in year 2030. And then cancer deaths will also increase from 8.2 million to 30 million annually. I never actually told this statistic to my wife, but men who are married are about 35% less likely to die from cancer than those who are not married. So I guess that there's another thing that I could thank my wife for. So as you see here, the cancer impact us financially, mentally, and also emotionally. So what about the oncologists in drug companies? According to FDA, new molecule entities, and then biologic license agreement approval, in year 2012, about 40 drugs are approved, and about 13 oncology studies approved. And year 2013, 27 drugs approved, and out of 27, about 8 is oncology studies, drugs. And year 2014, 41 approved, and about 9 is oncology drugs. And year 2015, 45 drugs approved, and 13 are oncologists. So as you see that from last 5 years, about 30% study drugs are approved by FDA. So now Big Pharma turned its focus to oncology because it will meet unmet medical needs. Actually, it will help the patients a lot. And there are a lot of dying patients out there. And then oncology drug is very profitable to a lot of pharma companies. And it becomes the cornerstone to their success. So according to global oncology revenue, in year 2015, Johnson & Johnson did about $4 billion, and BMS did about $4.5 billion. And Celgene did more than $8 billion, and Novartis their revenue is more than $10 billion, and then Roche Genentech did more than $25 billion. In industry-wise, there are about $107 billion the oncology revenue, and then it is expected to increase to $150 billion in year 2020. So basically, next 5 years, oncology revenue is going to increase about 50%. So what about as a programmer and statistician? What do we feel about oncology studies? First of all, it is very different from other therapeutic areas. We're going to talk more detail in next slide, but oncology has a lot of different data collection and data point and data analysis. So it becomes very complex, from building to clinical database system, in Caseform, SAP, and TFL, STTM atoms. So it becomes very difficult to study to do. So let's see how oncology study different from other therapeutic area study. First of all, there will be tumor measurements and their response to drugs. So we should collect those information and measure those information. In addition, there's oncology-specific measurement for response criteria, such as liver and spleen enlargement, bone marrow infiltrate, and blood count. And also, there will be oncology diagnosis measurement, including immunophenotype, performance status of ECOG, and staging information. And a lot of biomarker and other information as well, too. And drug administration exposure could be a little bit different. Nowadays, there's a lot of pill for oncology drug, but most of the time it's going to be inserted by the milligram per milliliters. So there's a different measurement for the drug exposures. And toxicity become very important for oncology study because a lot of patients are sick and they're in critical stage. So toxicity for the lab and adverse event become very important for oncology studies. Oncology study also has its own unique analysis for time to event analysis, such as overall survival, progression-free survival, and time to progression, and many other time to event analysis. And obviously, there is a different data structure for oncology studies, such as STTM, ADAM, and also control terminology that we're going to talk more detail in our later webinar. So in biometric department, we have a big challenge. Since there will be more oncology study coming to us, biometric department need to scale our team, our infrastructures, and also our knowledge. For example, actually a lot of our clients are struggling to find good oncology programmer and statistician because they're expanding really fast and they don't have easy time to find those people. Another challenge is that since oncology study is complex and difficult, it is challenging to how to prepare the biometric department so that programmer and biostatistician conduct oncology study more effectively. So with this oncology booming and a complex oncology study, usually the more difficult the challenge, the easier to distinguish the winners. Whether we will expand our department in biometric for oncology therapeutic area or conduct the pivotal oncology study for our company or sponsors. So the question is that, does your department want to be winners? Let's talk about the story. About a couple of months ago, I took my son to state middle school math contest. My son was selected to represent his middle school, even though he was still 8th grader. So he was basically competing against 8th graders. And when my son saw this problem, actually he started solving this problem by x equal 998. And actually there is a better way to solve this problem rather than x equal 998. When I saw how my son solving problem, he's just 6th graders, I noticed that it doesn't matter how smart he is. He's not going to win against 8th grader because he has not learned the easy way to solve these problems. What about in our biometric department? Can you imagine that programmer or statistician do not know much about oncology studies, about their data collections, analysis, but they're assigned to oncology study and need to just support and lead the complex oncology study? Are they going to be able to do their job more effectively, or they're going to succeed in the assignment? So how can you solve the complex problem? I mean, in this case, so in our biometric department, how can you make complex oncology study easy for programmers and statisticians? Usually, the way we solve the complex problem, we make it easy, either by equations, such as algebra for 6th graders, or methods, or trainings or mentorings. So now let's talk about how we could make oncology study easy for programmers and also statisticians. There are mainly 3 major oncology subtypes: solid tumor, lymphoma, and also leukemia. Solid tumor is abnormal mass of a tissue that are not cyst or liquid. And they are the most common cancers. And it is usually found in breast cancer, prostate cancer, lung cancer, liver cancer, and pancreatic cancer, and melanoma. Most of the study that we will do probably going to be solid tumor studies. Lymphoma is a cancer that actually starts in the lymph node. As you see in the right side in the screen, and this is a lymphatic system that goes into our body. And there's a lymph node that could actually collect the lymph and then spread the lymph. So lymphoma is the one that actually starts in the lymph node, and we notice it by that lymph node become enlarged. And also we could actually detect the nodal masses, such as the aggregations of the several enlarged lymph nodes. And also we could find extra nodal masses, lymphoma, that detected outside lymphatic systems. Leukemia is a cancer that usually starts in bone marrow and result in high number of white blood cells in your blood system. There are 4 major types, such as chronic lymphocytic leukemia, chronic myeloid leukemia, and acute lymphoblastic leukemia, and acute myeloid leukemia. In our presentation, we're going to focus about chronic lymphocytic leukemia. During the years, Oncology Society has created response criteria guidelines for the public, academic, drug company, and also research organizations. So in certain oncology study subtypes, public can refer to this response criteria and use it for what to collect and how to measure those collect data and how to determine response from those measurements. So solid tumor, RECIST, response evaluation criteria in solid tumor are the most popular ones. And for the immunotherapy, we also have immune-related RECIST 2009. And a lot of companies actually, for immunotherapy study, a lot of companies fail their clinical trial study before they actually implement immune-related RECIST 2009. So their drug approved. There are a couple drugs that are approved using that different response criteria. So selecting and the proper response criteria is very important for your clinical studies. Lymphoma also follow either Chesson 2007 and Chesson 2014, which is new response criteria for the lymphoma study. It is also known as 2014 Lugano classifications. And as you mentioned, the leukemia has 4 different study types. So the chronic lymphocytic leukemia, we follow IWC 2008. And then chronic myeloid leukemia, we follow CML, ESMO guidelines. And then acute lymphocytic leukemia, we follow NCCN guideline 2012. And then acute myeloid leukemia, we follow IWAML 2003. In our study, because we focus on chronic lymphocytic leukemia, we're going to discuss about IWCLL 2008 for leukemia studies. So solid tumor, which follow RECIST 1.1, actually collect target lesions and non-target lesions and new lesions. And then using all 3 information, the study or actually investigator or clinician determine the response for the patient at the given visit. The lymphoma study following Chesson collect tumor measurement by CT scan and tumor assessment by PET scan. The reason that we do PET scan is that a lot of time lymphoma tumor should be distinguished with the virus from the tumor. So that's why PET scan become important to make sure that that is a tumor. And also we collect bone marrow infiltrate and spleen liver enlargement. And then using all 4 information, clinicians or investigators determine the response for that patient at that visit. The leukemia following IWC era 2008 also collect tumor measurement by CT scan and bone marrow infiltrate and spleen liver enlargement and blood count, such as lymphocytic and hemoglobin and platelet. And using all four collection information, the clinician determines the response for the patients at that given visit. One of the most important aspects or factor for programmer and statistician in oncology study is oncology-specific CDISC data standards. About a couple years ago, CDISC STTM team introduced STTM tumor-specific domain, TU-TRRS, and also ADAM team developed time-to-event analysis dataset. This including the event descriptions and also censoring information for those events. And also control terminology such as response criteria in complete response, partial response, progress disease, and stable disease, and immune-related response criteria as well too. For the tumor measurement, terminology has longest diameter, sum of diameters, longest perpendicular diameters, area, sum of area, and tumor state. For the response, terminology include target response, non-target response, new region regressions, overall response, and best response. And there are a lot more terminology, but these are probably the one that we use the most. And then some of the terminology will be introduced, and also we're going to talk about in our webinar later too. For the CDISC STTM domain, there are three major tumor-specific domains. TU as a tumor identifications, and TR as a tumor result, and RS as a response. So let's talk about tumor identification STTM domain. It contains that tumor numbers, and also whether it's going to be target or non-target, and where it's located, and how you got measured, whether CT scan or MRI, and what visit. As you see that subject 001 has a 3 target and 3 non-target at the screenings. And this TU domain is very effective when you want to see how patients has responded to the drug, because it contains how many target tumors are found each visit, and also how those tumors, whether they merge together, or they split, or they increase, or some of the tumor disappears. So this TU domain kind of gives the investigator and programmer statistician see how that patient responds to drug in terms of tumor numbers. So now let's talk about tumor measurement. In oncology study, different subtype of the study determine how we could measure tumors. For the solid tumor, following RECIST 1.1, usually measure bigger than 10 million by CT scan. It is one-dimensional measurements. So in this example, we only measure longest diameter, which is 25 millimeters. And then we sum up all those longest diameter for the patients. If there are 3 targets, and we actually sum those 3 targets. In this example, there's a 3 target, target 1, target 2, and target 3 tumor site. And one is a 23, one is a 22, one is a 25. So we sum them up for that patient at the visit. So now it's a 70 millimeters, and this is going to be compared at each visit and determine whether how the patient or tumor respond to the drugs. Lymphoma and leukemia study has a really different tumor measurement. The size is going to be longest diameter is greater than 50 millimeters, and it is a 2-dimensional measurement. What they mean is that your product of the longest diameter and then perpendicular diameter. So 25 times 15 is 375. And we also some of those areas. So in this example, target what tumor has the longest diameter is 25, and longest perpendicular diameter is 15. So area is going to be 375 for this target 1, and then sum with the other target 2 and target 3. The sum of the area is going to be 1,060 square millimeters, and then this number going to be compared with other sum of the area in different visits. There are also 4 different response criteria, such as complete response and partial response and progress disease and also stable disease. In some cases, stable disease also same as non-progressive disease or non-complete response. So how are we going to determine the overall response at any given visit? We determine the response for the target and non-target and new regions. And then we're going to determine how is overall response for that subject at the given visit. For example, let's say target is a complete response and non-target is a complete response. And then there is no additional tumor detected. And overall response is going to be complete response. But target is a complete response, but non-target is a stable disease, and it's going to be partial response. Another thing we have to notice is that if any progress disease, for example, the target, there is the sum of the diameter is going to be increased from the baseline, then it's going to progress disease, or there's any non-target lesion increase, there's a partial disease, or any new tumor detected, it's going to be partial disease. So if any progressive disease, then everything is going to be overall response is always going to be progress disease. So let's look at in our STTM data set. At that cycle 1 visit, our sum of diameter for 3 targets is decreased to 35 from 70 from the baseline. So now it becomes partial response. And for the non-target, there was a 3 in baseline. Right now, there's also 3 in cycle 1. So there's no difference. Nothing disappear and nothing increase. So this can be stable disease. And we notice that there's no additional tumor detected. So it's going to be no. So using this information for target, non-target, new regions, overall response for the subject 001 at visit cycle 1 is going to be partial response. Oncology study also has its own specific analysis. It's not only used in oncology, but oncology uses this analysis all the time. We call time to event analysis, such as overall survival, progression-free survival, and time to progression, and many others. And also CDISC Adam team developed time to event Adam data set that we're going to talk a little bit later in our presentations. Overall survival, probably the most important and most popular endpoint in cancer studies. It is from the time from randomization date until death. It is the most reliable cancer endpoint, and it is a most time primary endpoint and peripheral endpoint. It is done in usually randomized control studies. So let's look at how we could create the time to event Adam data set, in this case for overall survival. So this subject ID and sex and treatment groups, and then primary is going to be overall survival as a days. And then this AVAL is going to be created from the start date. In this case, start date is going to be randomization date. And Another state is going to be event date. So AVAL is ADT minus start date, randomization date. And then event description also include for all the subjects. For example, subject 001 has a complete study, and 002 lost to follow, and 003 is death, and 004 is a progress disease, and 005 is ongoing. As you notice is that there is a sensor variable which determines what event is going to be censored or not. Since reading over survival, that is not censored, and all other event is going to be censored. Usually there are two major time to event analysis. One is a ROC-rank test and survival, and another is a Cox regression model. ROC-rank test, as you see in the code, that we're going to select the parameter as overall survival, and we're comparing by the AVAL, so time from randomization date to death, and then we're going to censor all those events excluding the death, and then we're going to compare between by treatment. But in some cases, we notice that some of the analysis depending on other factors, such as sex or age groups. In this case, we're going to use the Cox regression model. As you see that we're adding another variable, such as sex in the model, and then this regression model is going to be determined by treatment group and the sex groups in further studies. The most popular result for time to event analysis is Kaplan-Meier curve or Kaplan-Meier plot. As you see in this left side, the y-axis is going to be the probability of survival. Basically, how many patients are survived at given time. And x-axis is a time of observations. As you see that there is a drop from, let's say, here, drug B, from here, drop here to there. What they mean is that uncensored event happened, which means somebody died at that given time point. You also notice that there's a plus sign, which means it's uncensored, which means censored events, such as loss to follow, or complete study, or withdraw from the study. So those information censored, so it doesn't impact in terms of this graph, but only that's going to be impact, dropping the probability of survival. So the key point about this Kaplan-Meier curve is how wide these two graphs is going to be, and how consistent, and especially at that median level. And another example is a tables. Usually we create this table using the p-values. And then right now in this example, you could find this example is Adam ID for that time to event analysis data set. There are two methods we're using for this study, log-rank test and regression model. And according to those information, we actually produce a p-value and determine how two drugs are statistically different. So progression-free survival usually is a time from randomization to progressive disease or death. And sometimes it could be primary, but most times it's going to be secondary endpoint. And then it's going to be determined, it's usually done in randomization control studies. So Adam data set example for progression-free survival is very similar to as overall survival, except one thing. Right now, progressive disease is not censored. So in this example, this is the same data set that we have with the overall survival that we talked about in previous slide, and that is not censored, and progressive disease is not censored. But all other events going to be censored for progression-free survival. There are also other key considerations or checkpoints in oncology study, such as target and non-target determination. we didn't talk about and how some of the tumor become target and measure the tumor size, and some of the tumor is non-target, only detect whether it's present or not. So there is a way to determine, there's a response criteria to determine how those target or those tumor become target and non-target. And then in many cases, response should be confirmed. So even though even the subject 001 has a complete response at cycle 1, and a lot of times you have to confirm the response looking at the next visit, cycle 2, whether it's going to be really, really complete response or it might be something else. And how to determine best response for that subject. Subject has a complete response and partial response, but become post-disease for visit 1, 2, 3, and then what could be the best response for the subjects. And how to use those response for object response rate. Usually this analysis is done in phase 1 and phase 2 study, and sometimes it could be critical for that oncology drug become a breakthrough therapies. So this become very important, but we didn't cover for our presentation today. And nowadays, adaptive design for oncology study is very popular. And this adaptive design, you could do internal analysis during the study to determine whether you're going to drop the study or we could go for the study. So adaptive design is to help the company save the money and also company prepare their clinical study more effectively. We only talk about tumor and also response data collections and also STTM preparations. But there are a lot more other data collections, such as immunotherapy and also bone marrow infiltrate and blood counts and enlarged liver and then kidneys. So those information are also need to be collected, and also it should be prepared in a STTM data set. And a lot of time, the primary efficacy ADAM data set is very complex. So either you have a composite endpoint, time to event ADAM data set, and there is a certain way you could prepare those information more effectively. So let's kind of finish our story. So what is the easy way to solve the complex problem? I mean, this math problem for 6th graders. You could start using x equals 998, and you can use algebra to get this value really quickly. So yes, 6th grader need to learn about easy way, in this case, algebra equation, to solve these problems. Once he learned and knows how to use it, even 6th grader could compete with the 8th graders, because he could solve this problem within seconds, not minutes, because this problem is no longer complex for him. Using the equations and training, this problem become easy for the 6th graders too. The question is how we could make complex oncology study easy for the programmers and also statisticians. Just like school teach 6th graders on algebra, I believe we need to train and teach our SAS programmer on biostatistician oncology studies. So oncology studies are no longer complex, but it becomes much easier for them. Can you imagine how fast programmer and statistician do oncology study with the proper training, with the proper knowledge? And what kind of qualities? When I saw this in LinkedIn post, I really love this one. Let's say CFO asked a question to CEO, say, what if we train people and they leave? It is a very logical question from the CFO, because it costs money and time to train people. And usually they leave. They will leave some point. And the CEO response is this: what if we don't train them and they stay? Yes, training will cost money and time, but sometimes not training probably will cost a lot more than trainings. So let's finish our talk today. And yes, there will be a lot more oncology studies coming. As you see in our early slide, that oncology revenue is going to increase almost 50% within 5 years, which means that company is going to invest a lot more in oncology studies. And there will be a lot more oncology studies coming into our way. And then they are not easy. They are very complex studies. So we should make those easy for the programmer and statistician by training and introduce oncologists, response criteria, and specific data collections, and also oncology-specific data analysis. In order to help our client to make their life easy, CleanData also provide oncology training for programmer and statistician. It will be more detailed and customized training such as data collections, STTM, ADAM analysis with more example and use cases. We also provide CDS roadmap and implementation such as STTM, ADAM, and Defender XML, review guide, and company-specific CDS standard development, especially end-to-end standard-driven process for oncology studies. And we can also provide the e-submission package and also provide the oncology programming and statistics, provide high quality of biometric deliverable for oncology studies. CleanData is very active in social media, so you could find in our contact information LinkedIn or Facebook or Twitter, and I'm also very active in social media. So you could email to me as Kevin Lee at cleandatainsight.com or consulting@cleandatainsight.com, or you could find me either LinkedIn or Twitter. Molly?
Okay, thank you, Kevin. So at this time, I just wanted to remind everyone that we are recording this webinar today. So after today, it will be available on our YouTube channel, and there are two ways actually you can access our YouTube channel: the harder way and the easy way. The harder way is if you go onto YouTube and you actually try to search for ClinData Insight and find our channel that way. The easy way is if you go onto our website, cleandatainsight.com, scroll down where you find the link to our social media channels, and if you click on the YouTube one, it'll bring you directly to that. So we'll be posting this recording on our YouTube channel shortly after today, so that you can have access to that if you want to go back and review the material again. So at this time, if there are any questions out there, please feel free to type them in. Again, at the top left-hand corner, there's a question box that you can type your questions in, or you can, if you can't find that immediately, you can also type it into the chat box at the lower left-hand corner, and then I can read off those questions to Kevin. So let's give a few minutes for you to jog and type that in. But let's start with this one first, Kevin. How can we find oncology-experienced biostatisticians or programmers?
Yeah, actually, that's a good question. A lot of time, I kind of mentioned in our presentation that a lot of time company is struggling to find really experienced and programmer statisticians. And the way that you could find is that you could actually have to ask the question about oncology-specific questions, such as response criteria, STTM domain, or ADAM dataset, what could be censoring, what could be event, and also what could be oncology-specific analysis, such as overall survival and progression-free survival. And we also, the CleanData also provide a technical interview for those candidates, so we make sure that those candidates really know about oncology studies.
Okay, great. So knowing the right questions to ask. The second question we have is, can we put some of data, such as bone marrow results, in RS domain?
Actually, that is also another good question, and we don't recommend putting other data collections in RS domains, except that sometimes RS domain could have in some study that some companies, I remember that some study has two different response for local clinician response assessment and also central clinician response for the assessment. But beside those response criteria, we recommend people to put other data information into other STTM domains. So bone marrow is going to be probably LB and FA might be my choice.
Okay. Another question we have, can you share the challenges faced in ADAM?
Yeah, ADAM is that a lot of time-driven ADAM data set is I probably provide a very simple ADAM data structures, but a lot of time it's much easier to create intermediate ADAM data set. What they mean is that you create another ADAM data set before creating the overall survival or primary endpoint ADAM data, so that you notice what could be done, what could be traced before you create a final ADAM data set.
Okay, so should we put all the time to event data such as OS, PFS, or TTP to one efficacy ADAM data set?
Usually, we don't recommend putting all the efficacy endpoint in the one ADAM data set because it becomes huge, and then when you make a changes in one parameter, you probably need to change everything, and you have to run the program, all of them, for that related to that ADAM data set. So over primary endpoint, always one parameters. And then secondary endpoint, maybe you could add in couple and things like that, but putting all the information in the one egg probably is not a good choices to do so.
Okay, so is PE the right domain for spleen and liver data? And
yeah, actually.
And I think the other part is, and pardon me if I don't get this correctly, example SUV max. I don't know if I'm reading the questions correctly. Is PE the right domain for spleen and liver data? For example, SUV max is what I'm reading.
Yeah, I'm not sure about SUV max, but spleen and kidney information are usually scored is probably better into FE domain, because a lot of time the doctor physically examined that part. It doesn't done by, so they make sure that it is enlarged or not. So physical examination, that's why it is go into the P domain. But in terms of how many infiltrated, because you have to also you have to measure how much is infiltrated by percentage. So a lot of time it's much better to P domain and then using the FA domain to determine how many percentage is infiltrated.
Okay. Can you share key factors and logics you use in defining clinical benefit response for solid tumors?
So clinical benefit response, I'm not sure how to answer that question, and I think I probably need to get back to about that one.
Okay. So I guess they can email you at klee@clindatainsight.com if they want more clarification or additional questions. Okay, so can we have oncology study following two different response criteria?
Yes, it could happen. I think that a lot of times you determine how you actually, what response, because what response they're going to follow, and then what response they have to do it. And so you have to determine in the either inclusion criteria or diagnosis, because some patients going to be lymphoma, and some patients going to be leukemia, some patient could be solid tumor. So yes, some cases, I see that some study actually have a couple different tumor types, and so they have to follow two different response criteria. So it could happen that way.
Okay, so one last question I have here. Should all the studies follow public response criteria?
No, actually, oncology study is sometimes, the study specific. So even though they follow response criteria for certain subtype, study oncology subtype, but they could also change because your study. And I noticed that some analysis or some specific oncology doesn't fall into those subtype or response criteria. So company kind of change their own response criteria from that existing response criteria. So those information is going to be pretty much company and study specific.
Okay, so I think that's all that I have right now. I don't see any other questions coming in, but if you think of any questions coming in after we sign off here today, please feel free to contact us. You can reach out to us via our website. You can contact us via consulting@cleandatainsight.com or klee@cleandatainsight.com. So at this time, I'd like to thank all of our participants for joining us today, and thank you, Kevin, for being our presenter. And I hope everyone has a good day. Thank you.
Thank you, guys.